So a quick summary. Who will be giving a quick summary? Devangi or Jania? Anyone would like to volunteer? Yourself? Quickly, people. Devangi, what about you?
Yes, sir.
Yeah? Okay, great. Thank you.
So we have two types of body fluids, blood and lymph,
Mm-hmm.
and they basically circulate in our body to transport certain substances. So blood is a liquid tissue and a connective tissue, and it flows in vessels, closed vessels.
Okay.
Whereas lymph is present in tissues. So blood is also called a tissue because it has different type of cells that come together to form a common function. And the fluid matrix of blood is known as plasma. So blood consists of 55% of plasma and 45% formed elements like WBC, RBC, and platelets.
Okay.
Plasma is a straw-colored and viscous substance and constitutes 55% of the blood volume. And the components of plasma are 92% water, 6 to 8% dissolved proteins like fibrinogen, albumin, globulin, and small amount of minerals like sodium ion, calcium ion, and it also consists of glucose, amino acids, and lipids.
Yep.
So the fibrinogen protein present in plasma is used for clotting. Globulin is used in the defense mechanism and immunity, and albumin is for osmotic regulation.
Yes. Okay.
Then we have a type of plasma called serum. That is plasma without clotting factors, means the fibrinogen are absent. And the reason why blood doesn't clot inside our body is that because inside the body the clotting factors are inactive.
Right.
Then we started formed elements.
Yep. I think you can all see the screen, right?
Yes, sir.
So my video is spotlighted for you. So any one of you who wish to keep the video on while the class, I encourage and appreciate that. You can do so. Yep. Yes. Continue.
So the first formed element is erythrocytes, also called RBCs. They are biconcave in shape, and we have 5 million RBCs per millimeter cube in our blood. And the pigment called hemoglobin is present on the surface of RBC that contains iron core and transports oxygen and carbon dioxide to the cells.
Yes, correct.
In males, we have 12 to 18 grams of hemoglobin per 100 ml of blood. And for females, the normal is 11 to 15 grams per 100 ml.
Correct.
And RBCs have a lifespan of 120 days, but they do not spend more than 50 to 55 days in circulation, because after that they reach an organ called spleen or the graveyard of RBCs where they are destroyed.
Correct.
And mature RBCs do not have nucleus.
Yes. So I think, did I ask you that why they do not have nucleus and what is the advantage? And did I give you this question?
Yes, sir.
I suppose, yeah. Anyone figured out why RBCs do not have a nucleus? And if they do not have a nucleus, how are they surviving? Like, what is the advantage? It has to be a planned thing that RBCs do not have a nucleus, right? In biology. What is the benefit of not having a nucleus? Anyone figured out?
Yes, sir.
Yes, please tell me. What did you figure out?
It is because it enables them to contain more oxygen carrying hemoglobin.
How? Yeah, that is a good, that's correct answer. But do you understand the logic? Why, if you have a nucleus, why can't it contain more RBCs? Because more hemoglobin, because hemoglobin is anyway on the surface, correct?
Yes, sir.
It's not inside, so it's on the surface, and oxygen binds to RBC on the surface of the RBC. So what's the problem with having a nucleus inside? Is it a space problem? Not likely, right? Because it's not like you have to fill in the hemoglobin and the oxygen inside the RBC, so you remove the nucleus. So do you understand why? Or anyone would like to guess? So though they will not ask you, but this is one thing I wanted you to ask, to find logic behind such questions. Okay, so this is a carry forward homework. Then you have to figure out why. What is the advantage of RBCs not having nucleus? Mature RBCs not having nucleus. Are there any other cells in our body that do not have a nucleus when they are mature? Any idea? No. Okay, so please figure that also out. Just some searching for you, food for the thought. Okay, continue, Devangi.
So,
Yes.
so RBC also lack mitochondria as well, right?
RBCs, yes. Why?
Then how are they going to do their respiration?
Exactly, that's what I'm asking. RBC seems to be like an empty vessel, but it's still a very important cell in our body, correct? So mitochondria becomes a little complex. So let's not go to mitochondria yet, because if you'll tell me about nucleus, I will tell you about mitochondria. Okay. So I want you people to reach at this answer, logical answer. Just don't. Yeah. Yes. Tell me.
So most of the
Devangi.
Google sites said that just to accommodate hemoglobin inside them, they lose their nucleus.
No, that's why most of the Google is not correct. Only 10% of what's written in Google is correct, to be very, very honest. Correct in the sense like biologically and logically correct, because just a simple superficial search. So go to this is also a homework for you to learn which sources to trust and which sources to not trust. Okay, so you can go to particular resources, right? Even your NCERT comes from, there are websites for education board for biology. There are various institutes of biology which have their own website. So Google is a search engine. If you go to Google and search that I have a headache, what I might be suffering from, the first pop-up will be brain cancer, brain hemorrhage, but it might not be true. You just might have had a rough day, correct? So dig deeper, and whatever you read, think that, okay, is it making sense? Oh, it seems logical, or it does not seem logical. So knowing that, just to accommodate hemoglobin. So is hemoglobin inside? And if it is inside, how is it carrying oxygen then? Is oxygen going inside? If it is going inside, then it is the bound form, is it inside? Then how do cells take it? All these things we'll discuss once you'll tell me the answer, okay? Cool. So let's first finish what we have to finish today, then we can come back to this question anytime. Yes, Devangi, continue.
So can you scroll up?
Yes, can. Yep.
No, sir, down.
Here.
Second type of formed element, leukocytes. Leukocytes or WBCs, they are colorless and they do not have hemoglobin, and they have different types of nucleus as there are different types of WBCs. And normally in 1 millimeter cube of blood, we have 6,000 to 8,000 WBCs. So Types of WBCs are granulocytes. They have granule-like cytoplasm, and it consists of neutrophils, basophils, and eosinophils. Second is agranulocytes, that are lymphocytes and monocytes. So the
Yes.
most abundant WBC is the neutrophils, 65%, and the least abundant is basophils, 1%. Third is thrombocytes or platelets. They are the fragments of a big cell called megakaryocyte that is formed from the bone marrow, and they help in clotting of blood, and they bind together to heal at the site of injury. And in 1 millimeter cube of blood, we have 1.5 lakh to 4.5 lakh platelets. Then blood grouping. According to ABO blood grouping system, we have 4 different blood groups: A, B, AB, and O. A and B are the names of antigens. They are molecules present on the surface of RBC, and antibodies bind to antigens and destroy them. So in A blood group, we have A antigen, and because of that, the antibody is anti-B. And it means that the B blood group person cannot donate. So the donor of A group are A and O.
Perfect.
Then B group, B antigen is present and antibody is anti-A. So the donor can be only B and O. Then AB group is the rarest, in which both A and B antigens are present. And so there is no antibody. So all the 4 groups AB, A, B, and O can donate blood, and hence it is also known as universal acceptor, as it can receive blood from all.
Yep.
And the last O group is most common, and it has no antigen, but antibodies are anti-A and anti-B. So only O group can donate to O blood group people, and it is universal donor also.
Yeah, correct. Then we talked about RH group, right?
Yes, RH stands for rhesus factor. That is another antigen on the surface of RBCs. It was first figured out in rhesus monkeys. So it is 2 types, RH positive and RH negative. In RH positive blood, there won't be anti-RH antibody, but in RH negative blood, there will be anti-RH antibody because the RH antigen is absent. So basically, we can give RH negative blood to RH positive patient, but we cannot give RH positive blood to RH negative patient.
Yes. So now taking this into account, who's the universal acceptor and donor? Taking both RH and ABO blood grouping together.
So combining them, the universal acceptor is AB positive group and universal donor is O negative.
Yes, perfect. And then I ask you to go back and read about Erythroblastosis fetalis. Anyone?
So
Yes.
I have a
Yeah.
question.
Yeah.
So can you just scroll up the ABO blood grouping part?
Yes.
Yeah. So in last class, I asked you a question regarding blood group, and there were specific donors for a particular blood group, and also you said that antigen. I mean, if O blood group is donating blood to A blood group, then the A antibody in O will bind to E antigen present in A, and there will be reactions going on, but still blood donated to a person is 1/10th, around like 250 ml, and also we are not concerning over A antibody because giving blood to the person is worth saving the person, right?
Yep. Yep.
Yeah. And like considering the same fact, why cannot O blood group receive blood from others? I mean, if it's only 1/10th, then why cannot O receive blood from other?
Why can't O receive blood from A antigen and B antigen, right?
No, from
Sorry,
A, B, or AB
blood
group.
group A, B, or AB, right? So that is because that's a good question. That's a good observation you did. So when we provide antigens, okay, So antibodies, which means, so if I'm providing antigen into someone's body, so antibody in that body is more, right? It can be reproduced. So if you give A blood group, A blood to a O person, so that A, whatever amount of A you are giving, you see the amount of anti-A that is present in the O group, it is very high, right? So the limiting factor should be antibody. If the limiting factor is antibody, then the reaction can be, it will be very slow, and you can withstand it. For example, if I give a O group into a person's body, so what does A has? It has anti-B, right? The antibody is this, which is present in a blood group, correct? O does not have any antigen, but it has anti-A, which is very, very less, because the blood, which, so antibodies should be the limiting factor. But when you do vice versa, when you put A into O, there are lots and lots of anti-A, 5 liters of blood, and in that 5 or 4 liters of blood, there is lots of anti-A, much more than A antigen that is going to enter, right? Do you understand? Do you understand this point, Iram?
Yes. Yeah.
So what will happen is, the moment the body, the system will encounter A blood group, all the antigens will right away, within a very short span of time, very acutely, within the first half an hour, will be attacked and the blood will be destroyed. So it's not going to be a slow process that the system can accommodate with. You understand? Because antibody is in a very large amount, so they will all gather right at the place where you will start putting the blood in. So you have to put a cannula at some place in the arm from where you will put the blood in, right? Right from that moment, right from that entry point, all these antibodies present in O group will start attacking the blood. So you will have infection right away. Not infection, you will have a, what do you call it? You will have a reaction right away at that point. So that's the problem. But if you do the vice versa, where O group is entering A blood group's body, it is not entering with any antigen, because A group, what it has a lot of antibody, anti-B, but it is not entering with any antigen. It is entering with some of its own antigen against the A blood group, but it's not like that amount of blood, when it will enter, it will first mix and slowly go throughout the body. Yes, it will kill it will go and bind to some of the A antigen and start killing, but do you understand, that's a relatively slower process. 250 cc of blood cannot kill all the A antigens in a blood group. So it will happen, but it will be very slow, so body can cope up with that. Do you understand?
Yes, sir.
Yeah. Does that answer your question? Is there any doubt still?
No.
So to prevent such kind of immune reactions, antibodies should be limiting factor. They should be in small amount. So when we are doing this thing, antibody is in limiting amount, right? That's why we can do this. But when we are doing this process, A into O, antibody is not the limiting factor. It is in very high quantity. So immune response will be high again. So you know that higher the antibody number, higher will be the immune response. Make sense?
Yes.
Yeah. Okay, that's good. Great point. Okay, so I ask you to go back and read about Erythroblastosis fetalis. Did anyone do that? Did anyone read Erythroblastosis fetalis, what it is? So we'll start with this. So look at this one chart here, which I have taken from Encyclopedia Britannica, is the source. Now, this talks about Rh hemolytic disease, but instead of focusing on this, you just know that what we are talking about is Erythroblastosis fetalis. We call it Erythroblastosis fetalis because it is a problem that occurs due to mixing of Rh. Now, how it can mix? So you understood that we cannot put any antibody against an antigen when the antigen is already present, correct, in high amount. Now look at what happens here. Sometimes this kind of situation can develop during pregnancy, but not during the first pregnancy. So what happens is, let's say there's a father who's Rh positive, okay, and the mother is Rh negative. Okay, so when these two will have a kid during the first pregnancy, okay, so this is, let's say, the first pregnancy. It could also be the second. If it is the second one during which it is happening, then subsequent pregnancy will be suffering. So just for the sake of simplicity, let's say this is the first pregnancy. Now, if the father is Rh positive, one gene at least of Rh positive can come. So father can be Rh positive for both alleles, or father could be Rh positive for one allele and Rh negative for other. In both these cases, the father is Rh positive. Depending on which allele it is giving to the baby can have Rh positive blood inside a Rh negative mother. You understand that? That part is clear? So in this mother, the baby is Rh positive, okay, but the mother is still Rh negative. Make sense?
Yes, sir.
Yeah. Now, when delivery happens, so no problem with this pregnancy because mother is Rh negative, so mother does not, and this is Rh positive, so no harm to the mother or the baby. It's okay. Because placenta is in between. There is a membrane, there is a junction called placenta which prevents the mother's blood from mixing with the fetus blood. So there's a very common myth, you must all have heard of it, that we are made up of our mother's blood or our father's blood, right? Have you heard of this? That a baby is made up of the mother's blood or the father's blood, correct? It's a very common myth in society. Mother's blood never mixes with the baby's. Baby makes its own blood when it's developing, because placenta is there to keep the mother's blood away from the baby's blood. Okay, so that's how a Rh positive baby can develop into a Rh negative mother. Even the blood group of the baby can be different from the blood group of the mother, and that stays like that. Now, at delivery, what happens is, because many deliveries, especially during natural deliveries, the placenta gets broken, like it tries to come out, it scrapes off during delivery, and during that breaking down of the placenta, some of the blood vessels get ruptured inside the mother's uterus. Just 1 second. Excuse me. So some of the blood vessels might get ruptured, and some of the fetus blood can get mixed into the mother's body. Now, the Rh positive baby's blood cells enter the mother's blood. Now, what will happen if Rh positive blood cells will enter into a Rh negative blood? So mother's blood has never seen a Rh factor before. Now, suddenly it sees a Rh factor. So what will the mother's blood do?
It will make antibody against it.
It will elicit a response from the mother. So mother's blood will think, oh, there is something which is not ours. We don't know this thing because it was always Rh negative. It has no Rh antigen. Now Rh antigen is there. So mother's blood will recruit all the anti-Rh antibodies to kill that Rh antigen, correct? And it will do it within a few days. It will do it. But antibodies have a thing that they can be stored as memory. Whenever antibodies formed against anything, it gets stored as a memory. Do you understand, people?
Yes, sir.
Yeah. Now, in the second delivery, months later, so the Rh antibody is present in mother's blood now, which happened due to this mishap during the birth. So now mother's blood has anti-Rh antibody because of this encounter. Now, in the second delivery, what will happen? Second pregnancy, what will happen? If by chance, again, the baby is Rh positive. If the baby is Rh negative, no problem. But if the second time also the baby is Rh positive, just when it is developing as an embryo, the anti-Rh antibodies from the mother will enter, because they are antibodies, they can cross the placenta, they enter into the baby's blood and they attack the Rh positive baby's blood. Sometimes it depends on how vigorous is this reaction, how much antibodies present in the mother at that time. If it is less, which is not able to kill all the blood but can still make the baby severe anemic at the time of birth, because most of the blood gets destroyed. And if not anemic, then at the time of birth the baby is born and it gets jaundice and things like that. So there is some kind of a disease. But in some conditions, if mother's blood has a lot of anti-Rh antibody, it can even kill the fetus and cause miscarriage of the pregnancy. So that condition is known as erythroblastosis fetalis, because the erythrocytes, are affected in the baby. Is this clear to you? Is it clear?
Yes.
Sir.
Yes.
Sir, I have a doubt.
Yes, please.
You said that the mother's blood does not get mixed with the fetus or the baby's blood, right?
Yeah.
Well, then how the the food or the nutrition is going to be transferred to the fetus?
Yes, good point. So placenta is like a network, interdigit network. So some of the mother's blood come and baby's blood vessels come, and they form a network, mesh-like network, but they never merge with each other. They make a network. And because these are small capillaries, just like how does oxygen gets into your blood? You breathe in oxygen in your lungs. How does it get into your blood? The same is the answer. Simple answer, diffusion, right? Diffusion through concentration gradient. When we breathe in, our lungs have around 21% oxygen, which is the atmospheric concentration of oxygen, correct? And the blood has very low oxygen because all the oxygen has been used by the bodies till when the blood comes to the lung. I will teach you about that in today's class only. So wherever there will be high concentration of oxygen, from high concentration it will diffuse into low concentration, given that the membrane is semi-permeable. So similarly, the nutrition, which is glucose, oxygen, even the waste from the baby, which is carbon dioxide, gets diffused and can cross these membranes and get into the other blood vessel. But the blood cells, WBCs, RBCs, platelets, serum, that does not mix at all. Does that answer your question, Rohan? Rohan, does that answer your question?
Yes, sir.
Yeah.
Thank you.
Yeah. Great. So, and the evidence is that if mothers and baby's blood were the same, then whatever is the mother's blood group should have been the baby's blood group, but that never happens, right? But it can happen, but it can also not happen. Mostly it does not happen, because the blood group is decided by which antigen you are getting from the mother and from the father, and the combination of that will decide your blood group. Okay. So even a mother with a blood group A can have a baby with a blood group AB or O, right? Or even B. No, not B actually. Oh yes, even B can be. B is possible if the father is B. So you can do all that permutation combination yourself and see which, if father's blood group is one of the four and mother's blood group is one of the four, which blood groups are permitted in the baby and which blood group are not permitted in the baby. So there are some blood groups that can never happen. For example, if father is O and mother is O, can you tell me what blood groups baby can never have? It's the simplest. O means no antigen. Yes. A, B, or AB. All these three blood groups cannot happen. It's not possible in the baby if both father and mother are O blood groups. a father and a mother with O blood group can only give rise to a baby with a O blood group. That is also another reason that over time, over generations, O blood group will accumulate and more people will be O blood group. That's the reason that around 40% of the world's population is O blood group. Okay, make sense?
Yes, sir.
So if a couple with both with O blood group have a baby with AB or AB blood group, that's not a biological child. You can just know that through biology, and you can, that's an adopted child. So these kind of questions do come in exams, so just prepare that. Is erythroblastosis fetalis clear? So this happens because of Rh antibodies. Now this can be avoided. Can you tell me how can it be avoided? So what we do not want is a memory, like we do not want anti-Rh antibodies to develop in the mother and stay before the second pregnancy. So what can we do? We do not want mother to prepare any antibodies of her own, correct? No, Waziha. So during the first pregnancy, anything can happen. During the first pregnancy, so mother has to be Rh negative for erythroblastosis fetalis has to happen. If mother is Rh positive, not at all a problem. If the mother is Rh positive, then whether the baby is Rh negative or Rh positive does not matter. But if the mother is Rh negative and the first baby is Rh positive, then during delivery, if some of the blood gets leaks and enters into the mother's bloodstream, the mother's blood will slowly start developing antibodies against it, which we don't want, because these antibodies can in the second pregnancy go and start killing the Rh positive cells of the baby if the second baby is also Rh positive. So this condition is called erythroblastosis fetalis. It happens. So to avoid that, what we can do is right after the delivery, just to be 100% sure, right after the first delivery, before the mother's body detect Rh positive cells and start making antibodies, you administer anti-Rh antibodies into the mother immediately after the delivery of the first child. So those will go and kill the Rh positive cells if there are any. And because they are administered antibodies from outside, they are not formed by the mother's body itself, so they will not be stored for very long. They will be degraded and mother's immune system will never be activated. Okay, so it's like you spilled milk and you don't want your mother to see it, because then you are in terrible problem. So before she sees it, you clean it. You understand, people?
Yes, sir.
Simple. So if you do that, you can prevent mother's body from forming the antibodies which can stay in the body for a long run. So that's erythroblastosis fetalis and how we can avoid it. So if you would like to note anything from here, please do that, or should I move forward? Should I go forward, people?
So, like, they kill the child cell
Who killed the child?
in order to prevent this?
No, not the child is delivered. After the child's delivery, because the placenta gets torn apart, right? When the baby comes out, the baby has this placenta umbilical cord, right? So during delivery, sometimes the mother's uterus is tightly in contact with the placenta, and placenta has umbilical cord. So when you are pulling the baby out, this can get rupture, right? And it does get rupture because all of it has to shed out. So the uterus can prepare for the next pregnancy after some few months. So during this childbirth, if it gets ruptured, some of the baby's blood can get mixed with the mother's blood, right? We have to kill that blood from the mother's body, not the baby though we cannot kill, no? You understand? So after the first childbirth, just think of it like this. There was a child in the womb who spilled the milk in the womb while coming out. Now, if mother system get to know that the child has done something like unintentionally, it has spilled something that can cause harm to the next child. So you went into the kitchen, spilled the milk, then you came out, and your sibling went into the kitchen, and the moment he went into the kitchen, your mother entered. Who's going to get beaten? Assuming that there is no conversation to clarify. Who will be the culprit? Who will suffer?
The sibling.
The next sibling, right? So we have to prevent that. How can we prevent that? So you spilled the milk. The moment you come out and before your mother or anyone enters, you just clean it. So while going out, this baby spilled some Rh positive cells in the mother's bloodstream. So those needs to be cleared before mother's immune system can identify it and start
How is
making
that cleared?
antibodies. Put antibodies from outside. The same way how mother will clear. Mother's body will start producing anti-Rh antibodies. It's all written here, right? So anti-Rh antibodies attack the Rh positive cells and kill them. So before the mother's anti-Rh antibodies starts to produce, you inject anti-Rh antibodies from outside and kill those cells. So mother's immune system will never know that there were some such cells. Is it clear?
Yes, but so that antibodies won't affect the second child?
No, because they won't stay there for that long, because it's not the mother's own produced antibodies. They will degrade after some time. They are the antibodies we are putting from outside.
Okay.
Right? So mother's immune system does not know about them, right? A king can only know about the mercenaries that the king is sending into the kingdom, right?
Yes.
If I send a spy into some other country, the president of that country does not know about it. No, but both the spies will do the same thing, figure out something and eliminate the threat. That's what we want. We do not want the mother system to get activated and start sending antibodies. So we send antibodies right after the first birth, just to be sure that everything, even if there was a spilling, those will get killed before the mother's antibodies will prepare. And we can only have a memory of antibodies that our own body is preparing. The antibodies which are given from outside do their work and then go away. That's called passive immunity. For example, in snake venom, whenever someone gets bitten by a snake, we give anti-venom, right? Because then there is no time for we do not want to wait for the immune system to get active, identify the venom. By that time the venom will reach everywhere in the system and the person will die. But in case of corona, where we do not have any preformed antibodies against the virus, what did we do? We give vaccinations. Vaccinations help you to produce your own antibodies against the virus, right? Do you understand the difference between the two, people?
Yes.
Perfect. Okay, so let's go forward and let me also tell you about this. Yes.
I have one last doubt.
Yes, please.
So thinking about it now,
yeah.
If, for example, a mother gives birth by surgery, like through surgery, so there is less chance of the mother's.
There are less chances. Yes.
Yeah.
There are less chances of this erythroblastosis fetalis happening, because then we are not pulling the baby like naturally. So we make a cesarean cut, we take out the baby and we just cut the umbilical cord. But there also, it's not like there are zero chances, but the chances drastically go down in C-section.
Yes, sir.
Okay. Yeah. Okay, now let's talk about blood coagulation. In vivo means inside the body. So we read about fibrinogen, right? So this is, you don't have to look at this very dreadful schematic and get scared. So you all know about platelets, right? The thrombocytes. So these are the platelets or the thrombocytes. And what do thrombocytes do? they have, so thrombocytes are just, as I told you, they are cell fragments of a bigger cell called megakaryocyte, right? So these platelets have upon a tissue factor, which you don't have to remember. Even if you don't remember this, it's fine, but just know that platelets get activated whenever there is any injury to the body and there is a blood leakage or blood flow. So we need to coagulate the blood to heal the wound. Now this whole of this part is for is very advanced and is just important for entrances. You should know that there are a series of factors that work. So starting with factor, they are named like this: 7, 9. So 7 and 9 together activate TF7A and 9A. So they get activated into this. Then 7A and 9A go and activate the factor 10, which becomes 10A. So A means active. Inactive 7 and 9, active 7 and 9 goes and make the inactive 10 into active 10. Similarly, then these active 10 factor goes, and now this you have to remember for boards. So this cascade of factors in the body, in the blood, converts prothrombin into thrombin. So this is the molecule you have to keep in mind. Thrombin is a very important molecule in our body. Sorry, it's a, yes, very important protein. So pro means inactive form of it. So all the time our blood does not needs to be clotted. So when everything is normal, your blood is contained in the blood vessels, there is no injury, thrombin remains as prothrombin. Only when there is a tissue factor, there is any damage or anything that activates platelets, then this whole pathway starts, and prothrombin, after getting signal from the factor 10A, activated 10 factor, converts into thrombin. So this is important. Thrombin then makes fibrinogen, again inactive fibrin. Inside the blood, both thrombin and fibrin stays as prothrombin and fibrinogen. Any enzyme with gen on it, like pepsinogen, is an inactivated pepsin that has to be converted into pepsin. Okay, so fibrinogen gets converted or activated into fibrin, active form, by this thrombin. Okay, and thrombin got activated through this factor 10A. When fibrinogen becomes fibrin, then what happens? It stabilizes. All these fibrins are fibrous, they stabilize, go into the region where the injury is there, and they get cross-linked. So they make cross-linked mesh network, which is known as the fibrin clot. So in every wound, the first thing that happens is this fibrin clot that prevents the blood to keep flowing. So this is how blood clots or coagulates. Is it clear, everyone? Yes, no? Just look at this once. I'm just marking with red what things you have to remember. You have to remember it all starts with platelets, which upon any tissue factor, any factor that can activate the platelets, they get activated. Then a series of cascades start, a series of pathways starts. If you can remember 7, 9 and 10, okay. If you cannot, it's okay. For boards, it's not important. But factor 7, 9 activate the factor 10. When it's activated, it's called 10A, 10 activated. If you can remember, it's good that factor 10A converts prothrombin into thrombin. Then thrombin converts fibrinogen into fibrin. Okay, this is not very important, so you can leave this part. When fibrinogen gets converted into fibrin, fibrin can now go and produce cross-linked fibrin clot. Is this clear, everyone?
So factor thrombin and fibrinogen are secreted by the platelets?
Yes, prothrombin and fibrinogen are secreted by the platelets. Okay, so they have to become thrombin and fibrinogen to be active. That is done by certain factors. Okay, is that clear?
Yes. So in also NCERT it said that calcium ion is very important for blood clotting. How does that work?
Calcium, yes. So calcium is so many of these pathways require something called as cofactors for activation. So for example, you know that for photosynthesis, for that matter, or glycolysis, all these are different biological pathways. You will also see sometimes they write for this step you need Mg2+, magnesium. Similarly, for this blood clotting factor, Ca2+, calcium 2+, acts as a cofactor for these proteins to activate these proteins to do their function. That's why calcium becomes important. If you are devoid of calcium, then blood clotting also goes down, deprived of calcium. Okay, that part I have not included here because that's some details, but yes, you can know that calcium is important for clotting, iron is important for formation of hemoglobin. Okay.
Yes.
Yeah. Any other doubt, people? Just look at this pathway and tell me if there is any doubt.
Sir,
Yes.
can you just explain from fibrogen how, like, what's the connection between it and fibrin?
Yeah,
I didn't understand that part.
okay, so let me do one thing. Let me delete every change that I have made, and I will use a ink. Let's say, let me use, give me a color, tell me a color which is not on the screen already. Okay, that will help you. Let's say if I use purple. Okay, purple is not here. So whatever path I will follow, you have to remember that for your exam. Okay.
Okay,
So
sir.
it all starts with platelets. Okay, platelets are known as thrombocytes. Now platelets every time in our body are not activated, so they have to be activated into what are called activated platelets, obviously. Now this can be done by various tissue factors. Now that tissue factor could be anything released during an injury or something, or where the clotting factor is required. So this tissue factor tells platelets to get activated. Once they get activated, within these platelets there is this series of activation of different factors that begin. So what you have to focus on is that activated factor 7, tissue factor 7 and tissue factor 9, when they are activated, they both go and activate tissue factor 10 and make it 10A. Okay, and this 10A goes and activates prothrombin, which is present in the blood. In a pro means inactivated form, into thrombin, which is a now active form. Now it can do its function. Now the function of thrombin is to convert fibrinogen, inactive form of fibrin, into fibrin. Okay, so do you see this arrow? It is going and binding, going and activating fibrinogen into fibrin. Once you have fibrin in your system, this fibrin can directly help in making fibrin clots, a mesh-like network that forms fibrin clot. Is it clear now, everyone?
Yeah.
So only the parts that I have highlighted with purple, you have to remember that. Anyone has any doubts? Good to remember these two factors if you can. If not, just remember this 10A. So first comes thrombin from prothrombin, then comes fibrin from fibrinogen. Okay, and then fibrin makes the clot. Draw it in your notebooks, people. Let me know when everyone is done. So you don't have to worry about this amplification phase at all. So this is not going to be, so this whole part is not in your syllabus. In fact, even this is not a part of your syllabus. Okay, don't worry about this.
Done, sir.
Done? Everyone done? Shall we move forward now?
Yes, sir.
Okay, great. So this was about blood coagulation in the body. And now I will come to the human circulatory system. We also name it the cardiovascular system. Right? Cardio means heart, and vascular comes from blood vessels. So it is a system of heart and blood vessels, because blood in humans only flow within the heart and the blood vessels. So this kind of system, where you have a master gland from where the blood starts the journey and it only travels within some confined spaces known as arteries or veins or capillaries, together known as blood vessels, is called a closed type of circulatory system. So humans have a closed circulatory system, right? We know about closed circulatory system from evolution and different type of animals that we studied, right? Can you tell me one example of any organism which has an open circulatory system, and what is the difference between the two? Anyone, quickly?
Yeah, arthropoda.
Yes, arthropods have open circulatory system. Great. And what happens in open circulatory system that is different from closed circulatory system?
In open, the organs are directly bathing the blood, whereas in closed, they're just enclosed within the heart and the vessels.
Great. Yes. So the whole tissue, the whole body, different tissues are directly bathed. Okay, so there is no concept of blood vessels carrying it from the heart to the tissues and carrying back the blood. So heart, if there is a heart-like organ, it just directly pumps all the fluid into the body cavity. Okay, cool. But in humans, we have to study the cardiovascular system. So let's first focus on heart. So you can see this image here, right? Let me just make it a little big. I hope it's visible to all of you.
Yes, sir.
Yeah. Do I need to make it more big? Should I zoom it more, or it's okay? Is it visible?
It's okay.
Okay, great. So this is how heart, a cross section of heart looks like in human. So it's very different from what we draw hearts like, or we associate heart likes in symbolic form. But if you see, it's not that difficult also, or that different also. So because I can actually, just to fiddle up, do you see that it still holds the shape? Yeah, I used a very bad color, contrasting background, but I think this is the part of the heart that we see. Now you can see that. Can you see what I'm outlining as yellow? Yeah.
Yes, sir.
Yeah, but this is just a cross section. Okay, but the point is, what we miss is the very important part here. Now coming back to the biological heart, the function of heart is just one thing: keep pumping the blood. So it is like a pump. It's exactly like a pump that we use to, put water into coolers, or put water in water tanks, or to drain water from somewhere. So heart has no other function. the only function. It has some other functions about which I'm not like, it's not related to emotions at all. So like we say that heart has no functionality with our thoughts, emotions. It just is a pumping organ in the body, and it is very straightforward and simple. So let's go to the components of it. So what you see here, if I use a white, I don't know if white will be visible. Let me use green. I think green will be visible. So can you see there are 4 chambers? One is this part. Can you see this one? Then there is this second chamber. Then there is this third chamber. And then there is this 4th chamber, right? Everyone can see the green portions that I have marked?
Yes, sir.
Yes.
Yeah. Now the above 2 chambers, these 2 are called atrium or auricles. Okay. So we can see here is your right atrium. This will be better. So this is your right atrium or right auricle, and here it is marked the left atrium or left auricle. Then the below ones are called ventricles. So here is the right ventricle and the left ventricle. So humans have a 4-chambered heart, just like birds. So birds and mammals have 4-chambered heart. There's one exception. There is one reptile also that has a 4-chambered heart. You know that? Anyone remembers?
Yes. It's
Crocodile.
crocodile.
It's crocodile. Crocodile has a 4-chambered heart, where but other reptiles, how many chambers are there in reptilian heart?
3.
3. In amphibian heart?
3.
3 again. So amphibians and reptiles, they are very closely related. And fish heart?
2.
2-chambered. Okay, so fish is at the beginning of the vertebrates. So fish has a 2-chambered heart. Amphibians 3, reptiles 3. Crocodile has 4. It's an exception in the reptiles. And then comes birds and mammals. They have 4-chambered heart. 2 atriums, 2 ventricles. Now, do you also can you also notice that the walls of the atrium, which I have marked here, see, the walls of the atrium are very thin, but the walls of the ventricle, can you see they are very thick? Yes.
Yes.
Ventricles have a thicker wall, whereas atrium have thin walls. Why is that? Because ventricles have to generate more pressure. They have to, when they are the chambers responsible for pushing the blood or pumping the blood into the whole body, throughout the body. So they need to generate more pressure. That's why their walls are thicker. When they contract, they generate more pressure. Okay, and if they are thin walls, upon more pressures, the wall can get ruptured. So that's one differentiation between ventricles and atrium. There is another difference between ventricles and atrium. Atriums always receive the blood. Okay, so whenever blood enters into the heart, it enters into the atrium, either the left atrium or the right atrium. So keep noting these things. Okay, you have to write these things, whatever I'm saying. So blood always enters into the atrium, right atrium and left atrium both. And blood always leaves from the heart through ventricles, left ventricle and right ventricle. Is that clear? Now we have 2 atriums and 2 ventricles. Clear, everyone? Shall I go forward?
Yes, sir.
Everything is written? Okay. Now we have 2 atrium. Why do we have 2 atrium? Both receives blood. From where does the right atrium receives blood? And from where does the left atrium receives blood? So this is right atrium. Where is this receiving blood from and where is this receiving blood from? Anyone knows? Okay, so right atrium receives the blood from the body tissues. Okay, from everywhere in the body. Our blood, which is everywhere in the body, after the oxygen is utilized, it goes back to the right atrium through via veins. Okay, so we'll talk about veins later. So let's say I'm not putting this part here, but right atrium receives blood from all the body tissues. Where does left atrium receives blood from?
Lungs.
Yes, so this receives blood from lungs. Okay, and from body tissues. Is this part clear, everyone? Now also let's
Yes, sir.
make it clear, where does right ventricle sends blood to and where does left ventricle sends blood to? So they both are sending. So where is right ventricle sending blood? See, right atrium is receiving blood, and from the right atrium blood goes, let me use a white color. From this right atrium, blood goes down into the right ventricle. And then from right ventricle, where should it go technically? Where do you want it to go?
If it's coming from the tissue, then it's supposed to go to the lungs, right?
Yes. So right ventricle sends all the blood which it receives to lungs. Then what something is happening in the lungs, and we know that from lungs it comes into left atrium. From the left atrium, it goes down into the left ventricle. And now where should it go from left ventricle? To body tissues, correct? Is this part clear? Then from body tissues again come back into the right atrium. From right atrium goes down into the right ventricle. From right ventricle goes to the lungs. From lungs comes into the left atrium. From left atrium comes down to the left ventricle. From left ventricle goes into the body, and the cycle continues. Now why it is going to the body and why it is going to the lungs, that we will talk about later. But is this part clear, everyone?
Yes, sir.
Okay. Apart from atrium and ventricles, there are many things. So there's something called aorta. Okay, let's first talk about aorta. Let's first talk about vena cava. So vena cava is the major vein that brings blood to the right atrium. Okay, from here it's coming, from here, through this vein, it's coming into the right atrium. Okay, now it is a vein, and vena cava, can you see here, it has two bifurcations. One is called superior vena cava, and this one is called inferior vena cava. Because where heart is placed, if you know where is heart placed, somewhere around the middle of your thoracic cavity, slightly tilted towards the left. But there is body above the heart and there is body below the heart. So blood has to come from both, from the brain, head, neck, shoulders, also lungs also, and blood also has to come from the abdomen, legs, hands, etc. So the part of the body which is below the heart, that blood comes through inferior vena cava. Finally, like it merges into inferior vena cava, and blood from upper part of the body comes from superior vena cava. They are called inferior and superior just on the basis of their positioning. One goes up, so superior, the other goes down, so inferior. Both of them merge together here and make the vena cava, okay, the bigger one, and then dump the blood into the atrium. Similarly, aorta is the, so vena cava is the largest vein in the body, and aorta is the largest artery in the body. Okay, also the most strongest one, because you see, aorta receives blood from the left ventricle. Okay, from the left ventricle it goes into the aorta here. So this diagram is not very clearly, it's a real like blood, a real heart's cross section. So to see things, I'm just clarifying the pathway for you. So from left ventricle it goes into the aorta, and from aorta, these bifurcations that you are seeing, that is because blood has to go to various different tissues, right? So one of it will go to the upper body, the other will go to the abdomen, the lower body, then will again bifurcate to go into arms and the legs, everywhere. Okay, but you can see aorta, how big is it, right? It's a very big artery. Yes or no?
Yes, sir.
So aorta and vena cava is clear, right? So vena cava brings into right atrium, aorta takes from the left ventricle. Two things are left, right ventricle and left atrium. Now left atrium, as I said, it also receives blood, but it is receiving blood from the lungs. Now for what purpose did blood went into the lungs, or for what purpose do blood goes into the lung?
To get oxygenated.
To get oxygenated, which means before going into the lungs it was not oxygenated, or it was, what happened?
Deoxygenated.
It got deoxygenated. Where and how?
It got deoxygenated in the tissues.
In the tissues. So which means that it was already deoxygenated when it entered the heart, right, from the vena cava, correct?
Yes.
Then it went into the right ventricle. From right ventricle it went into the lungs. So if it went into the lungs from right ventricle, what kind of blood left the right ventricle? Oxygenated or deoxygenated?
Deoxygenated.
Deoxygenated, correct? So a deoxygenated blood left the heart. So what will carry that blood? Veins or arteries? Whenever something leaves the heart, what carries the blood? Arteries or veins? How do you define arteries and veins? Anyone? Because I just want to change that knowledge. Can someone define arteries and veins for me?
Yes, sir.
Yes.
For veins, the vessels which supply blood to the heart, they are veins, sorry, and the vessels which take blood away from the heart are arteries, right?
Arteries. You must also have read one definition in junior classes saying that veins carry deoxygenated blood and arteries carries oxygenated blood, correct?
Yes.
Yeah, but that's not always true. What's always true is that veins bring the blood to the heart and arteries take the blood from the heart. Okay, because here what we are seeing is that from the right ventricle, what kind of blood is leaving the heart?
Deoxygenated.
Deoxygenated. But what kind of blood vessel will carry it? A vein or an artery?
Artery.
An artery. So and what is another difference between veins and arteries? Have you studied that veins have valves, arteries do not have valves? Veins have a thin wall and arteries have a thicker wall of their membrane, correct? And always
Yes.
the pressure. So when we see the pulse, what are we checking? A vein or artery? When you check the
Artery, right?
pulse, yes, you're checking an artery, because blood pressure is higher in artery. It's pulsating in the arteries but not in the veins. So in the arteries, you do not have to worry about backflow, because the pressure from the heart is so much that it will keep pumping. But when it goes into the veins, it is collected back by the veins and it goes back to the heart. There the pressure is not there, so you need to depend on valves. Valves prevent the backflow. So veins have valves, arteries do not have valves. So from from right ventricle, and also whenever in human physiology I'm saying right of an organ, it is right, not when you are looking into the organ, it is the physiological right. So you might have noticed that the right is written on the left side when you are reading the book, right? Or you're reading the screen, so it's towards your left side, but you have to imagine it, the heart which is inside your body, correct? So if this heart is inside your body, this is the right ventricle, correct? Yes or no? It's not a mirror image. So from right ventricle, blood goes into the lungs and it goes through an artery known as pulmonary artery, which is this one. So here, this one, pulmonary artery. Pulmonary artery is called pulmonary because the word is associated with lungs. So pulmonary arteries takes the blood from heart into the lungs, and write down, it is the only artery in the body which carries deoxygenated blood. That's why it's color coded as blue. Okay, so pulmonary artery is the only artery that carries deoxygenated blood from the heart to the lungs. So if I make another link from this right ventricle, you can see it's going here. Can you see this, people? My arrows?
Yes, sir.
And this will go to the lungs. Okay. Now, when it goes to the lungs, it gets oxygenated. Oxygen enters into the blood, CO2 comes out of the blood capillaries. Now the blood is oxygenated. So from lungs, when it will come back through pulmonary veins. Okay. These two here are pulmonary veins, which are, why there are two? Because there are two lungs from where, again, here also it's two. It bifurcates. One goes into one lung, one goes into other, then again it bifurcates. So pulmonary veins are the only veins in the body that carry oxygenated blood. So these two are exceptions. So through pulmonary veins, the blood comes into the right auricle. So it comes here, and then from here, let me make it a little better. So blood comes here, and then from here it goes down, and from down it goes into the aorta. Clear, everyone? So pulmonary veins carry oxygenated blood and pulmonary arteries carry deoxygenated blood, but rest everything remains the same. Pulmonary artery does not have valve, pulmonary veins can have valves. Pulmonary artery is thicker, pulmonary vein is thinner, and pulmonary artery takes blood away from heart, pulmonary veins takes blood towards the heart. The only difference is between oxygenated and deoxygenated blood.
Sir?
Yes.
Sir, earlier you said that it's not always the case that veins carry deoxygenated blood and arteries carry oxygenated blood. Do you say so because of the pulmonary veins that carry oxygenated blood?
Yes. So that is the exception. So pulmonary veins are the only veins. That's what I dictated, right? Yeah. They are the only veins that carry oxygenated blood, and pulmonary artery is the only artery that carries deoxygenated blood. That's why you cannot just say that all arteries carry oxygenated blood, because pulmonary artery does not. You also cannot say all arteries carry, all veins carry deoxygenated blood, because pulmonary vein does not. But these two are the only exceptions. Okay, but yes, there are exceptions. Rest all other arteries and veins in the body apart from pulmonary carry oxygenated and deoxygenated blood respectively. Is that this part clear, everyone?
Sir.
Yes.
Actually, what's the use of valves?
Yes, we are coming to other things. We have just finished aorta, vena cava, artery, veins, pulmonary arteries, pulmonary veins. I'm coming to other things.
Okay, sir.
Okay. Now there are other things marked here. One is this SA node, sinoatrial node, this thing. Okay. Then there is this AV node, atrioventricular node. Now, if you look at the location, I will be using a different color. Let's say purple. So one location is here. Purple is not visible. Which color should I use? Should I use yellow? Yeah. So one location is here, right? This is for the SA node. Other location is here. This is for the AV node. What are these two nodes? As the name suggests, sinoatrial node. So this node is some specialized heart tissues, heart cells that generate the, heart contracts. How does heart contracts? Heart beats, it contracts and relax. It keeps doing it throughout your life. In fact, heart is the first organ that is formed and is functional in an embryo. On the 22nd day, just after 3 weeks of implantation, you will see that there is a functional heart. It will not be the heart, a complex heart like this, but after the 22nd day, there are 2 beats that you can also put a stethoscope on the mother's womb and see a slight, very feeble beat. So heart starts beating, or heart tissue starts beating. So SA node is called the pacemaker of the heart. These tissues of SA node, they produce a slight current, which, when by current I mean electric current, a real electric current. There are 2 kind of tissues in our body that works through electric current generation or electric potential generation. One is neuron and the other is the heart. So this SA node generates some current which gets passed on to the AV node, atrioventricular node here. So first the atria contracts, first the auricle contracts, followed by the ventricles. So if you look a contract, if you see the videos of a contracting beating heart, first the atriums contract and then the ventricles contract. Okay. And when the ventricles contract, the atriums relax. And when the atriums are contracting, the ventricles are relaxed. Do you understand, everyone? So SA node produces current, electric current, which is important for the muscle contraction, sends it to the AV node. AV node sends that current throughout the heart. Can you see this yellow part going from the AV node to all different regions of the heart, all the walls of the heart? In between is the septum. So this part here, this region here is the septum, cardial septum. It divides the left heart from the right heart. And this yellow thing that you are seeing is called bundle of His. So these bundle of His, technicality, but this bundle of His is a tissue which comes from the atrioventricular node and spreads that electrical current throughout the heart, so that when the atrium is done contracting, the next thing is the ventricles that will contract. Is these things clear? And then this is the interventricular septum. Apex is the part of the heart which is towards which the heart has this out bulge. So the bulging of this heart. So this bulging side is called apex. And then there are valves. Now coming to valves, there are valves. So blood is going from atrium to ventricle. It never mixes from one atrium to another atrium and one ventricle to another ventricle. That is a problem. If there is a hole in the interventricular septum, you must have heard that this person has a hole in the heart, or that person had a hole in the heart, right? Have you heard this or not?
Yes,
No,
sir.
sir.
No, never heard of holes in the heart?
No.
No, sir.
Never watched Kal Ho Na Ho movie, a classic Shah Rukh Khan movie. He had a hole in the heart. So there are two type of holes. One is in this interventricular septum. If the septum is not properly formed, that's called the hole inside the heart, between the left and the right chambers. If there is a hole, then always oxygenated blood, which is present on the left side of the heart, always gets mixed with the deoxygenated blood which is present on the right side of the heart. Okay, so these person will be lethargic, they will not be very, so they will have symptoms as they grow, and they will be less and less efficient in energy or oxygen availability. So with this kind of a hole, the person can still survive after birth for some time, and with a small surgery you can get this fixed if it is not a very major or a bigger hole. Another kind of hole is in the membrane of the heart itself, like in the wall of the heart. So the blood is leaking out of the heart. In that condition, the baby or the fetus cannot survive, and oftentimes, a child stillbirth or a dead baby is born, or just after the birth it dies in a few weeks if not treated. And it's very difficult to treat those kind of holes. Okay, so intraseptal hole is less lethal and can be treated. Okay, so this septum keeps the oxygenated blood away from the deoxygenated blood. Now, when blood goes from atrium to ventricle, I told you that first atrium open, blood enters, atrium contract, all the blood goes down to the ventricle. This is happening simultaneously for both the atrium. When all the blood goes into the ventricle, next thing that has to happen is the ventricles to contract. So when atriums contract, valves open. So between the right atrium and right ventricle, there is a valve known as tricuspid valve at this junction. So let me use yellow again. Here, which I'm marking, here is a tricuspid valve, and right here is a bicuspid valve. So you should write it. Between the left atrium and the left ventricle, there is a valve called bicuspid. Bi means two, cuspid means two flaps are there in that valve. How many of you have seen classic or like Bollywood movies where these police stations used to have these hanging doors, like two doors like this hanging? You understood what I'm saying?
Yes, sir.
That's like a bicuspid valve, two valves that open and close. Bicuspid valve is also known as mitral valve. MITRAL, mitral valve. And then between the right atrium and the right ventricle, there is a valve called tricuspid valve. So it has three cusps, which means three flaps. Okay. Is it clear, everyone? Bicuspid and tricuspid valve. The whole function of the valve is same. Function of valve everywhere in the body is same, to prevent backflow. So when blood from atrium went into the ventricles, now the ventricles will contract, and ventricles have to send blood. The left ventricle has to send blood through aorta into the whole body, and the right ventricle has to send blood through the pulmonary artery into the lungs. But if these valves are not there, when the ventricles will contract, the blood can just go back to the atriums, right? Yes or no?
Yes, sir.
So to prevent that from happening, we have valves in place. Okay. And then these valves should be fixed and should be sturdy in place, and that happens due to these muscle fibers called chordae tendineae. Chordae tendineae. So these chordae tendineae are strong muscle fibers that keep the valve in place and does not allow the valve to, displace or remove or broken. Okay. So there are chordae tendineae both in mitral, in tricuspid valve, and also here in bicuspid valve. So is the whole heart clear to all of you?
Yes,
And
sir.
also, this is a figure from NCERT which I want you to go back today and look and try to understand. We discussed everything about the schematic plan of circulation, how from right atrium it goes to the pulmonary artery, right ventricle it goes to the pulmonary artery, to the lungs, comes back and all. So just look at this. And this is where I end today's class because we are ahead of our time. In the next class, I'll be teaching about this pathway. So this is known as a dual circulation, a double circulation that is happening, and the ECG. So please read about this part from NCERT, which we just discussed today. I showed you, I explained you. In the next class, I'll tell you about different layers of the heart. Then we'll go to double circulation, and in the end, we will study the ECG, electrocardiogram. You all must have seen something like this in movies, right?
Yes, sir.
Yes. So what is this? What does this signify? What information can we get from this thing? We will study in the next class. Okay.
Okay, sir.
Any doubts, people? Anything you want to ask? If
Sir, do
no,
you
then
have
yeah.
a different SA node for the left atrium?
No. SA node is same. SA node is just at the at this place. What do we call? The sinoatrial node is just near the right atrium. From the right atrium, the left atrium is just adjacent. So through this bundle of his, it can just go to the right atrium. Okay. To send it into the ventricles, we need a AV node, atrioventricular node. So we don't have different SA nodes for different atriums. Okay. We also don't have different AV nodes for different ventricles. There is just one AV node and one SA node. Then they bifurcate the signal because it's electrical signal. It's just like putting a wire in the house. You can send the current wherever you have the wirings. Okay. But first comes the SA node, then comes the AV node. You must have heard of pacemakers. Some people have surgically implanted a battery near their heart, right? Pacemaker. Anyone knows about pacemaker?
Yes, sir.
Pacemakers. What do pacemakers do? Why do so in the people who have a faulty SA node, so the electric current cannot be produced, their heart goes arrhythmic. So the rhythm of the heart, that heart has to beat every 0.8 seconds. So every 0.8 second heart should beat. That's how we know that 72 beats per minute is the normal beat. So every 0.8 seconds, your SA node gives a small impulse of current and the heart beats. If this is not working properly, then your heart becomes arrhythmic. Now, SA node's functionality can be modulated when you are doing exercise. It beats faster, so it produces more impulses. When you are sleeping or you are conscious, unconscious, subconscious, it goes low. So that can be modulated. But normally when you are awake and sitting, it should be around 0.8 seconds per beat. But if that goes faulty, then people need an artificial machine which has wires, literal wires, encoded at the right place where the SA node is, and that gives impulse. So that machine gives impulse, electrical impulse, every 0.8 second and keeps the heart beating. It has a battery. It's battery operated. Okay.
So the person will not die if AV node is not working. Sorry, SA node is not working.
Yes, the person will die if SA node is not working. That's why you need to put a pacemaker. So SA nodes is not like if it stopped working right away, like right at the moment, then your heart will stop beating at all, and you will get a cardiac arrest. But in people, in older people, you first see the symptoms. Their heart is arrhythmic. They will feel the symptoms. They will go to a doctor. Doctor will do a ECG and tell you, okay, your SA node is weak. It's going to be non-functional soon. So you better get a surgery done and put a pacemaker in your chest. Okay.
From the chest, how does it initiate the impulse?
It's a current. So from the chest there are wires going into the heart. It's just implanted above the heart in the chest region. So there are literal wires going into the heart tissue at this SA node region, and this device, which is pacemaker, creates an electric current every 0.8 seconds, sends an impulse every 0.8 seconds. Pulse transistors, electricity, anyone? So there are many devices that generate current in a rhythmic fashion, correct? Okay.
Yes, okay.
Yeah. Cool. So I'll see you all in the next class. Take care. Bye-bye. Devani has one more question before we leave. Why there's a possibility of backflow of blood in veins but not in arteries? Because in veins, the pressure of blood is low because veins collect blood from the tissues and tissues are not pumping it into the veins. Arteries collect the blood from the heart